Whoops. Alright, hello everyone, and welcome. Whoa, sorry about that. Jeez. There we go. There we go. And welcome to your last review for Biol1140. Today we're going to go over the last few units that we've discussed in class, such as transcription, translation, inheritance, sex-linked inheritance, monohybrid, dihybrid cross, that sort of thing. So let's just jump right into it. So, right now we're going to start off with something known as the central dogma of biology. Now, why is this so important? What is the central dogma of biology? And before I continue, if you hear anything in the background, that's likely just my cat. He's kind of messing around with my stuff currently, but that's okay. So, what is the central dogma of biology? Basically, if we simplify it down to its parts, what it's essentially saying is that from DNA, we then get RNA, right? And then from RNA, we get proteins. And then from proteins, we get all of our gene expression. So DNA is expressed through proteins, but DNA has to be first transcripted into RNA, which then has to be translated into a protein. So, what do these terms mean, right? So just think of what the word transcription means on just a um, basic language level, right? Transcription essentially means that I'm going to write something down, like let's say hello. I wrote it down here, right? So if I'm going to transcribe hello, I would just basically do it like this, right? That's transcribing something. I mean, if you hear like the prefix trans means to like change something, right? So and scribe means to write something down. So transcribe, we're changing what we're writing, kind of. If that makes sense. We're transferring what we're writing to something else. It's do a different medium, if you will, right? So translating, right? If we're translating, we're changing how we interpret something, right? So that would be like taking hello and turning that into something like, I don't know, hola, right? So this, our translated word can now be interpreted by something or someone else, right? That's basically the central dogma of biology. We have an entire genome. We have all of our DNA. We have all of our DNA that exists within us in each and every cell. We have all of this, but we have to only but in the case of most cells, we only have to express just a small portion, and that small portion can change from cell to cell, right? But the point is that we go from DNA to RNA to proteins. We get from DNA to RNA via a process called transcription, and we go from RNA to proteins via a process called translation. Right? That's a very basic level of understanding it. So moving on then, let's just go over what transcription is, right? So we went over DNA replication, right? A couple of units ago, I think. And basically what that was saying is that we have a few different proteins, a few different enzymes that come into DNA, unzip it, and essentially make a duplicate copy of DNA. Now here, we're going to do something kind of similar. So let's draw our DNA, right? So now this is our DNA that's been opened up, and let's draw our nucleotides, our bases. So let's say this one is 5 prime and this one is 3 prime. Right? So remember, an important note for any sort of replication or transcription, an important note is that it's always synthesized from 5 prime to 3 prime. So now I've had a couple of questions of what this means. So let's look up a quick nucleotide. I guess it's not really going to show up. Okay, well, let's try this. Okay, well, that didn't really work how I wanted to at all. Anyway, let me see if we can find a better picture. Okay, here we go. This is actually better. So, here's a nucleotide, right, on a molecular level. So notice we have the nitrogenous base, the sugar, and then the phosphate group. And we number our carbons on the sugar because this is a pentose, right? So in this case, this would be deoxyribose, right? But this is a pentose. Pentose meaning 5 carbons. So we have 1 carbon here, and we go clockwise, right? 1, 2, 1 prime, 2 prime, 3 prime, 4 prime, and up here 5 prime. So basically, when we're adding DNA nucleotides or RNA nucleotides in the case of transcription, we're always going to add this phosphate group, and we're going to add it down here to the OH on the 3 prime end. Right? So that's basically what we mean when we say 5 prime and 3 prime, which you don't need to know this specifically. It's just good to know in terms of what we're talking about. So, of course, DNA runs anti-parallel, meaning that they are parallel to each other, but they run the opposite directions. So we have the 3' end on this side and the 5' end on this side. So basically, what we're going to do is we're going to have something known as a RNA polymerase come in. Now, just let me look up a quick picture of this, just to make sure I'm not giving you anything wrong. Okay. So basically, an RNA polymerase kind of looks like this, right? It's a big bubble that travels across the DNA that it unzips. So the RNA. Sorry, let me step back a moment. RNA polymerase does the job of like 3 enzymes that normally would do its job during DNA synthesis, right? So RNA polymerase takes the place of a DNA polymerase, a helicase, and a primase, and a ligase for that matter. So basically, this giant this giant enzyme is just going to go along and do its thing, right? So we're reading from 3 prime to 5 prime, right? We're reading along here. So we're reading down here, for example. We're reading, and then so now what we're going to do is we're going to add complementary bases. So we start 5 prime here, right? And then we're elongating it in the 3 prime direction. Right? So this moves along here, 5 prime to 3 prime. Right? We have the 5 prime end here, 3 prime end here. We're reading from 3 to 5. We're making from 5 to 3. And then we're going into the DNA. We're unzipping as we go, and that's a function of the RNA polymerase. We're making DNA as we go. Sorry, we're making RNA as we go. RNA is in red here, right? So, transcription has a few different steps to it. So these steps these steps are initiation, elongation, and termination. Right? So keep this in mind later because we're going to revisit these three steps exactly later on, right? So remember this. So basically, initiation is exactly that, where RNA synthesis starts. So let's say so this is going to be downstream. This is going to be upstream. So let's say upstream over here we have a sequence of DNA. I guess it would be up here or down here or whatever. We have a sequence of DNA where the RNA polymerase is going to recognize it. The RNA polymerase is going to see that and say, "Okay, that's where I need to bind." And it's just going to and then it's going to bind and then it's just going to keep on transcribing, right? So that's the process of elongation, right? We're elongating the RNA. We're making a polymerase. No, sorry, no, we're not making a polymerase. We're using polymerase to make a polymer, right? We're elongating this RNA polymer so it could be something that we could use later, right? In this sense, we are literally transcribing what the DNA says, right? So now this RNA is going to go on and make a protein, right? And we're taking the DNA, we're taking this region here, and we're going to turn that into a protein, essentially, through the use of RNA, right? So that's elongation. And then termination is, let's say, all the way down here, there's a region of DNA. There's a region of DNA in which it says, okay, now go to the RNA polymerase. Just release, go away. I don't want you anymore. That's what the termination sequence is. So termination sequence is when transcription stops, and that's a sequence in the actual DNA. So we have the promoter region here. Sorry, I forgot to mention that. This purple region is the promoter region. Promoter region influences the RNA polymerase to bind to DNA. And now the RNA polymerase is going to elongate the RNA. And then as we go along the DNA until we stop, we hit a terminator sequence in which RNA polymerase then leaves. So that was the basics of transcription. Now that we've covered transcription, let's go into translation. So in the case of eukaryotes, the RNA, our newly formed RNA, is going to go out. It's going to go out and go into the cytoplasm, right? So we're doing this in the nucleus, right? And it's going to go out and go into the cytoplasm. In bacteria, you don't need to do that, right? In bacteria, we transcribe and translate at the same time, right? So, just real quick. Okay, so now we go into transcription, where we're going to now utilize the mRNA to make a protein. So for this, there are also 3 steps. And if you'll notice the following. First step is initiation. Second step is elongation. And the third step is termination. Okay, well, didn't we just go over this? Exactly, we did. Right? These 3 steps are exactly the same. Right? Translation and transcription have initiation, elongation, and termination sequences. However, how they differ is in how the processes actually take place. Of course, we have to initiate transcription, we have to elongate the mRNA, and then we have to terminate where the RNA polymerase is. In the same sense, for translation, we have to initiate translation at the ribosome. We have to elongate the amino acid chain, and we have to terminate where the amino acid chain is going to be finished translating. Right. So this process occurs at the ribosome. So let's draw out the ribosome. So the ribosome has two subunits. We have this top subunit and this bottom subunit. Now, ordinarily, these two subunits are separated, right? They're um disconnected from each other, right? That's normal, right? However, when we have, I guess let's fill them out. Why not? Oh, didn't want to do that. So that's how they ordinarily fare, right? Just separated like that. So now, when we go on and take our mRNA, our mRNA now binds to this bottom region. This is the initiation of translation. So over here we have, so over here in this region, this is like a non-coding region, and over here this is also a non-coding region. We have to look for first our stop codon, in which that would be AUG, always, right? AUG, right? So AUG, once we hit that, then that initiates translation. So now we have our top subunit here, and now we can begin. So now, translation has begun, and we have the ribosome in its fully formed self, right? So now we could divide the top subunit of the ribosome into three different sections. We have the A site, the P site, and the E site. A for accepting, P for processing, and E for ejection. And it's at these sites that we have something called tRNAs. Now, what are tRNAs? tRNAs are RNA sequences that kind of look like this. Now, let's use it in blue. They kind of look like this, and they have what's known as an anticodon. So let's draw the anticodon in green. They have what's known as an anticodon here, where this portion will bind to each codon on the mRNA. And we have, okay, let's draw this in a different color. And we have our amino acid here on the top. So this is the anatomy of our tRNA. And if you're very curious, we could go ahead and see an actual rendition of a tRNA. Because I know my drawings don't always do it justice. Okay, well that one's low quality. So this would be a tRNA, right? Like it kind of has these loops here, and then we have this bottom loop with the anticodon, which binds with the actual codon, and then we have this end sequence in which the amino acid actually binds to it, right? So that's our tRNA. So basically, our tRNA is going to go ahead and bind to the A site. I'm sorry, this is very crude. You have to excuse me. Okay, let's shrink it down. Yeah. So this tRNA now binds at the A site, at which it's just going to go right into the P site, right? It's just going to go right into P site, and now this anticodon is interacting with the actual codon, where it's being processed. And now we then have a different tRNA at the accepting site, and now what could happen here is that this amino acid can then transfer over to here, thus elongating it. Okay, well, hold on. Sorry, I'm having a bit of trouble here. Thus elongating it, right? So let's go ahead and erase this. I can't really see where the eraser is, sorry. Right, so that's what happened. Oh, well, so yeah, so that's what's happening at the processing site, where we're actually elongating this amino acid chain. I think I'm wrong about this actually. Hold on, give me 1 second. No, yeah, that's right. That's right. Okay, so basically now, what's going to happen is that this is now going to go into the ejection site. This one is going to take its place, and now our amino acid chain is going to transfer over here. And now this tRNA goes on and leaves. That's why it's called the ejection site, right? And then we have another acceptor site, and then our amino acid eventually elongates and elongates, right? We go on and we go on, and then we have a stop codon. So now this stop codon has a release factor in it, what's known as a release factor, right? So now this doesn't have an amino acid because it doesn't code for anything. What this is now going to do is it's going to go here at the acceptor site. It's going to tell the ribosome, "Hey, it's time. We're done here. We're finished." Just and as a quick example, let's say it'll be here at the end. Like I think UAA is a stop codon. Once we read UAA here on the ribosome, UAA codes for the stop codon, and the stop codon has a release factor which tells the ribosome it's time to stop translation, right? So now our amino acid chain is released. Both tRNAs leave as well. And then the top ribosome subunit detaches, and then the mRNA goes on and leaves. So now we have a finished amino acid chain, also known as a polypeptide, also known as a protein, which can then go on and express itself in the cell and further in the body. So initiation, start codon, elongation, EPA site where we're elongating amino acids using tRNAs, termination, stop codon where we then go back to normal. So that was transcription and translation. How are you guys feeling? I mean, I can't really hear you through the screen, but I hope that was enough of an explanation, right? So through this, right, through the production of proteins via this method, we could say that this is gene expression. The production of a gene product through the expression of DNA to RNA to proteins. That's gene expression, right? So now we go further into gene expression. So now we go into the Mendelian model of gene expression, right? So now with Mendelian genetics, we have to know a few different terms: dominant, recessive, homozygous, heterozygous, codominant, incomplete dominance, and I think there's one I'm missing, one that's really important. Allele. And yeah, we'll figure out more as we go along. So let's start from the bottom. Allele is kind of the most important one. So remember, in humans, we have two sets of chromosomes, one from dad and one from mom. Now, when we put together these chromosomes, we refer to those we refer to those as homologous pairs, right? Homologous chromosomes. So that's what we're doing here. So now these chromosomes have the same locations of the genes. They have essentially the same genes. So we have this region here that codes for hair color, and on the same chromosome, on its counterpart, the same region will also code for hair color. And over here, eye color. Over here, eye color. Over here, height, that sort of thing, right? And those are just some obvious examples of gene expression, right? Basically, these two chromosomes determine how the body expresses itself, right? So remember, we get one from dad. Sorry, we get one from dad, and then we get one from mom, right? So at one point, these two chromosomes existed in a gamete. So if you remember, gametes are, of course, sex cells, right? And I just draw the chromosomes like X's here because it's the most familiar way to draw it. But these once existed in gametes, so we can go ahead and draw a sperm cell. It has the DNA in it, right? And then we can go ahead and draw the egg cell, which is much larger, right? So we also have the DNA in here, right? So of course, once a sperm fertilizes the egg, now we have a full set of chromosomes instead of just one set. So both of these would be considered haploid cells. So if you notice here, I drew 4 chromosomes in each one. So this would be n equals 4. This would be n equals 4. So that means we have a total of 4 chromosomes in each one. So 4 chromosomes here, 4 chromosomes here. And now, when we go ahead and combine them, right? When we go ahead and combine them, the sperm releases itself into the egg. Now we have a full set of chromosomes, right? Now this will be 2n equals 8, right? We have, so if we divide this by 4, or sorry, basically what this is saying is that we have 8 total chromosomes, and we have 4 pairs of 2. That's basically what it's saying. That's homologous chromosomes versus, just normal chromosomes anyway. So when we say we get one from dad, one from mom, that's what we mean, right? Because those gametes at one point had a complete set of chromosomes because they were at one point in diploid cell, and then through meiosis they had to split apart, and now we have two different sets of chromosomes once we have a fertilized egg. Two gametes coming together to make a full set, right? So now, that was a bit of exposition, right? So now we have these different points on the chromosomes. Now these points code for certain things, right? As we saw earlier, DNA to RNA to proteins, right? Now it depends on what is going to actually be expressed here, right? And this is where dominance versus recessiveness comes into play, right? So let's say this gene locus, and that's what's called a loci or locus, right? This gene locus codes for hair color, or let's use eye color as an example, actually. This gene locus codes for eye color. So now dad has brown eyes, and mom has blue eyes. Now, when we're talking about dominant versus recessive, the dominant trait, if it is completely dominant over 1, the dominant trait will be fully expressed. Always. Right? So if mom was also had brown eyes, then So this is the geno. So this is that's a couple of other ones. Genotype and phenotype. So where was I? Right. So okay. So both of these came from mom and dad, right? However, so this is the genes of an individual. So this is one person. So people can't have a brown and a blue eye, right? Or they don't have a mix of both. Some people do, but that would be a mutation, right? Now, in this case, brown will be completely expressed over blue, right? Blue will not be expressed at all because brown is completely dominant over blue. In this sense, at this allele, at this gene loci, we can say that this is a dominant allele and this one is a recessive allele. So we can say capital B brown here and lowercase b blue here. So now when we combine these two, we get what's known as a genotype. So genotype referring to the combination of alleles in your chromosomes, right? In your genes. The combination of alleles in your genes. That's your genotype, right? So here we have a heterozygous genotype, meaning that at one point, well, no, meaning that the genes in which the genes that are expressed, sorry, let's step back a moment. It's been a bit of a long day, so you have to bear with me. So, what I'm trying to get at is when they're heterozygous like this, it's essentially referring to the fact that both alleles at the same locus are different. So we have this locus here, we have the allele that's dominant, and then we have this one here, the same one, and the allele is recessive, right? So when we combine these two, we then get a genotype that is fully dominant. So this will only code for brown eyes, right? Only code for brown eyes. Another important thing to know is that once this individual makes sex cells in their body, then we can completely separate these two, right? Because that's what happens during meiosis, is that they separate each other, and this is known as the law of segregation. Is that during anaphase we have a pair of homologous chromosomes, we have a set, and then during anaphase these split apart into different gametes, right? So now we have one gamete with the fully dominant genotype we have one gamete that's fully dominant at this loci, and we have a gamete that's fully recessive at this one. So now this comes into play with inheritance, right? So if we go further with this, so now if we go further with this individual can have gametes of either capital B or lowercase b, right? So if we cross this individual with someone who also is heterozygous, now we could form something known as a Punnett square. So this individual can also have two types of gametes, one that is fully dominant or one that's fully recessive. So once we cross these, we can form a Punnett square. And now a Punnett square essentially tells you the possible gamete combinations in in an offspring of two individuals, right? So here we have capital B, lowercase b. This is one parent. And down here we have capital B, lowercase b. This is the other parent, right? So now we just cross like multiplication. Just like that, right? So let's actually write it out. Capital B, in this one we have capital B, capital B, capital B, lowercase b, capital B, lowercase b, lowercase b, lowercase b. So all of these are the possible allele combinations, all the possible genes of the offspring at this locus, right? So if we have two people with brown eyes, both are heterozygous, we therefore have a 25% chance of it being doubly dominant, a 50% chance of being heterozygous, and a 25% chance of being doubly recessive. So now notice how I say doubly dominant and doubly recessive. Now there's a term for this, and I've wrote it down over here, right? This is referred to as homozygous versus heterozygous. So hetero means different. Homo means same. Think of it like homosexuality or heterosexuality, right? Homosexuality means attracted to the same sex. Heterosexuality means attracted to the different sex, right? In the same sense, over here we have one capital, one lowercase, so they're different. One capital, one lowercase, they're different. These would both be heterozygous. So these would both be heterozygous. I will write that with red here. But now, this one has capital B, capital B. So now we can consider this to be homozygous. But look over here, this one has lowercase b, lowercase b. This one's also homozygous. So how do we make that distinction? Well, capital B will be homozygous dominant. Lowercase b will be homozygous recessive. So they're both still homozygous, right? They're both still homozygous. But the terminology but the second word reflects like the actual state of being, if that makes sense. So dominant versus recessive, both are homozygous, however. So now these are just the genotypes. So when we refer to homozygous, heterozygous, we're mostly just referring to the genotypes. When we get into the phenotypes, now the phenotype is the actual physical manifestation of how a person's genes are expressed, right? So the genotype is the combination of alleles. The phenotype is how those alleles are expressed, right? You can think of them as the actual physical traits. So pH, right? Phenotype. Same thing with physical. Physical starts with a pH, right? So think back to the one of these parents. These parents have a capital B gene and a lowercase b gene. But remember, this lowercase b gene isn't expressed at all. So the phenotype, the genotype is going to be heterozygous, but the phenotype will always be brown because we still have the dominant gene present. So therefore, on this Punnett square, we can then say that 1, 2, 3 out of 4 will be dominant. I guess we could write that in red. We'll have the dominant phenotype. And then 1 out of 4 will have the recessive phenotype. So this, what I just drew here, is what's known as a monohybrid cross. A monohybrid cross. Where we're crossing, where we're detecting the hybrid of two people and their possible genotype combinations. And with this case of a fully dominant, of two people who are fully dominant, sorry. Two people who are, okay, let's step back. A basic monohybrid cross, this typical monohybrid cross that you see here, will be between two people, both of whom are heterozygous at the allele that we're testing, and both alleles are controlled by complete dominance. So we'll get into incomplete dominance and codominance later, right? But for now, this is your basic monohybrid cross. We're crossing one allele, one gene, right? In this case, eye color, right? And if we have both that are heterozygous, right? So here we have two possible gamete combinations. Here we have 2. 2 times 2 is, of course, 4, right? 1 second. Sorry about that. If we cross these, then the ratios will always be 3 to 1. 3 dominant phenotypes to 1 recessive phenotype. 3 brown to 1 blue. So that's a basic monohybrid cross, right? So now, what if we wanted to cross or okay, well before we go into a dihybrid cross, what if we had this? Homozygous dominant crossed with homozygous recessive. So here we only have one possible gamete on the recessive, and here we only have one possible gamete on the dominant. So therefore, we could simplify the Punnett square to say this. So 100% of the offspring will then have this genotype, capital B, lowercase b. 100% of the population, 100% of the offspring population will have brown eyes. So just one moment. Okay. So yeah, so we could say this is the parent generation, and this is the F1 generation, and then the offspring of the F1 generation is the F2 generation. So at P meaning parent, F1 meaning first filial, first child, and F2 meaning second filial, second child, or second generation in this case. So if we cross these two parents, then we get the F2 generation. If we cross these two, the parents, we get the F1, right? So now, moving on. So now moving on, let's go into a dihybrid cross, right? So what if we wanted to test for 2 genes, right? What if we wanted to test for 2 genes? Let's use the example of pea plants, right? So let's say we have let's say yellow is dominant to green, so that's color, and then round is dominant to wrinkled, that's shape. So we'll say color, let's use Y's, and then shape, we'll use R's. So here, now remember, we're testing for 2 genes now. So now we have 2 genes represented in each of our examples, right? Or even easier, how about this? Right? So for this case, let's say this is the parent generation. To simplify this, because we're gonna expand this out to 16 boxes later, right? So this is a dihybrid cross, where we're crossing 2 genes, right? So this one is doubly dominant, this one is doubly recessive. I guess quadruply dominant, quadruply recessive. So remember, yellow is dominant over green, and then wrinkled is dominant over, sorry, brown is dominant over wrinkled. Right? So this is a wrinkled green pea, and this is a round yellow pea. So we're going to cross these 2 peas with each other, these 2 pea plants with each other. So this is the parental generation, right? So look at here. We only have one possible gamete combination. Y with R. Same here. Y with R. That's our only possible combination. So then we can now cross. Okay. So let's draw out a Punnett square. So we can now cross these two with each other, right? So we have this. So and then we have this. So now we have our possible combination of this. That's our only outcome. 100% of the outcomes will be this, right? So if we crossbreed this with itself, because plants can auto fertilize. Now we have the makings for our actual dihybrid cross. So now here we can make 4 possible gamete combinations for either parent. So of course 4 by 4 is 16, right? So we're going to draw 16 squares later on. So our possible combinations are capital Y, capital R, capital Y, lowercase r, lowercase y, uppercase r, lowercase y, lowercase r. And the same thing for over here, right? We have 4 possible combinations. And these combinations are going to be so these combinations are gamete combinations, correct? Right? So we remember what I meant when we say that, right? So these gamete combinations, we're not going to have YY in one gamete, and we're not going to have RR in one gamete. We're going to have YR in one gamete. So then when we cross these gametes with each other, then the final offspring will have the proper amount of genes, the proper amount of chromosomes, right? So what will this look like? Basically, so let's expand this. Let's actually write this out, right? So let's see here. All right. So now we have our possible combinations. Capital Y, capital R here. Capital Y, lowercase r here. Lowercase y, capital R here. And lowercase y, lowercase r here. So these are all of our combinations for one parent. One parent we got over here, right? Now this side will be the other parent, right? So let's do it again. I actually have an easier way. YR here, or sorry, capital Y, capital R here, capital Y, lowercase r here, capital lowercase y, capital R here, and lowercase y, lowercase r here. Now, what do these mean? It means that this gamete has double dominant, this gamete has one dominant, one recessive, this gamete has one recessive, one dominant, and this gamete has double recessive. And when we cross these, we'll get a distinct ratio that we'll then discuss. So let's fill it out, right? Let's just go ahead and cross it now. So you won't be expected to do this on the exam, but it's good to see where this comes from, right? Sorry, this one's wrong. Yeah, it could get very easy to overlook certain gamete combinations. Now, it doesn't matter how you orient your Punnett square, but I highly suggest that you do anyway, or you orient it in a way that's understandable to you. Typically, we orient our Punnett squares exactly how I've drawn here. Sorry, oh my gosh. So now this is our fully filled out Punnett square. So now let's see where we're at here. So let's pay attention to where our dominant genes are. So here we have a dominant gene. I'll with a yellow circle. How about that? Because that'll be green. Sorry, that'll be yellow and round, right? Yellow and round here. Yellow and round here. 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 Whoops. Here. 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 And here. Right? So now I'll say star is wrinkled. So we have a yellow star here, yellow star here, yellow star here, and then we have green round, right? So we have green round here, green round here, green round here, and that leaves only one possible combination left. Green star. So these are your possible phenotypes from this combination. So let's look here, right? We have 1, 2, 3, 4, 5, 6, 7, 8, 9 that are double dominant, that have double dominant phenotype. We have 1, 2, 3 that are dominant on one gene, recessive on the other. We have 1, 2, 3 that are recessive on one gene, dominant on the other. And then we have one gene here that is recessive on both. So this ratio is what's known as a 9 to 3 to 3 to 1. 9 to 3 to 3 to 1. So 9 out of 16 will have double dominant phenotype. 3 out of 16 will have one recessive. 3 out of 16 will have one recessive, one dominant, and 1 out of 16 will have double recessive. That's what this is referring to. So I can't see you guys right now, so I hope that this makes sense. Because yeah, we're gonna move on because this is quite tedious. You will have a couple of questions regarding this ratio, so it's really important that you know the ratio, but it's not as important that you're able to actually draw the Punnett square out. So yeah. Anywho, so now we're gonna move on to incomplete dominance, right? So let's say, so for the case of incomplete dominance, let's say we have a red flower and a white flower. So now this red flower has a genotype of capital R, capital R. The white flower has lowercase r, lowercase r. So when we cross these, oh, whoops, sorry. When we cross these, we get one possible genotype. However, this genotype, capital R, lowercase r, so this is we heterozygous, right? This genotype actually codes for pink flowers, right? So now when we cross pollinate this, we get R capital R capital lowercase lowercase lowercase, right? So 25% red, 50% pink, and 25% white. So what does this mean for the genes itself? So when we're referring to incomplete dominance, it is essentially saying that one gene, or we so okay, so if we have our homologous chromosomes, right, and we have the locus here, it essentially means that the gene at the locus does not exert full dominance if there is a quote unquote dominant gene. So it does not exert full dominance because the recessive allele is exhibited as well as the dominant allele. So in this case, if we mix white and red, as I'm sure you learned from kindergarten, if you mix white and red, you get pink, right? So if we have the double dominant red and the double recessive white, if we cross those together, we then get pink. And if we cross those together, we then get this ratio. So this is no longer 3 to 1. This ratio is now 1 to 2 to 1. 1 red to 2 pink to 1 white. So that is incomplete dominance when neither gene is completely dominant over each other. So both genes are therefore expressed in the case of a heterozygous genotype. So now we're going to go over codominance. So for the case of codominance, I like to use blood, right? So remember, we have 4 blood types: A type blood, B type blood, AB type blood, and O type blood. And then when we get in the case of positives and negatives, we're not going to go into that right now because it doesn't pertain to this lesson. So when we have AB, and O, what this is telling us is that there's something else at play other than just the typical Mendelian genetics of like dominance and recessiveness. In this case, we actually have 2 dominant alleles that are at play. The so let's say this let's use I as an example. So I is still capital, but now we're going to use the superscript A for the case of A type blood. Capital I B for the case of B type blood, and then lowercase i for the case of O type blood because it's still recessive to these two, right? So in this case, right, we have a few different we have a few possible combinations, right? So whatever in here is just, the gametes, right? So now if we have an actual genotype, A type blood can look like this, or it can look like this. B type blood can look like this, or it can look like this. O type blood can only look like this. Now, a B type blood, now this is the case of codominance, where both A and B types are exhibited. We have IA and IB. So let's look at this example. Hopefully this hopefully this actually works. Yeah, that's good enough. Okay, so here on A, we have these antigens. Antigens essentially act as like flags to detect foreign objects within the bloodstream. So if the blood detects, okay, well this sorry, if we have another blood cell, okay, well this has the same antigens, it has the same flag, so this we're all good. But if it detects a B type blood, then we have they conflict. So now A type blood is going to go ahead and attack B type, right? So here now look at AB type though, right? AB type exhibits both the B and A antigens, right? So now AB type blood can actually accept any blood, right? If you have a blood transfusion, for example, this can accept any type of blood. As for O type blood, now O type blood has no antigens, right? So with this, it doesn't actually have a problem mixing with A or B or AB. Hence why it's known as the universal donor, and why AB is known as the universal acceptor. Because O type blood, if we try to mix B with O type, well, those don't actually mix. Or A with O, those don't mix either. But vice versa, then they actually mix, if that makes sense. I don't know if it did. So going back to this example, right? We have AB type blood, B type, A type, and O type. Sorry. So with this, we can therefore say, right? We can therefore say that there are three blood type alleles found in the human population: A, B, and O. Or dominant A, dominant B, and recessive. But each human only exhibits two alleles at a time, right? So if we actually cross these, right? If we actually go ahead and cross, so let's go ahead and cross someone with A type blood, someone who's heterozygous for A type, and someone who's heterozygous for B type. So when we cross this, now we get the following, right? Look at this. So now we get lowercase I here in this box, A, B in this box, B in this box, and A in this box. So with this, we could therefore say that this ratio is 1 to 1 to 1 to 1. Because each possible phenotype outcome is actually different. A, B, B, A, and O. So let's quickly go over sex linkage as well. So remember, sex linkage, men have XY, women have XX. So it's the Y chromosome that determines whether or not something is male versus XX, which is just female, right? So if it could be XXY or XXXXY, it'll still be male because it still has that Y chromosome, right? So we then use the example of color blindness, right? So if we use the example of color blindness, so let's say X superscript. I don't know if I could do superscripts in Microsoft Paint. Never mind. So So this is color blindness, right? Color blindness is an X-linked gene, right? It's an X-linked allele, and it only exists on the sex genes, right? Because we're here at the sex genes, right? And we have the Y chromosome here, which it doesn't carry this gene. So for men, we only have the X chromosome that can carry the gene. So color blindness is therefore much more common in men than it is in women, because for men you only need to have it on one gene, versus in women you need to have it on both. Right, so for this case, if we actually cross these, so let's say capital B is normal vision, lowercase b is color blindness. So if we cross these, so you can see this woman is still has normal vision, but she's a carrier for the color blindness gene, right? So now if we cross these, we have a 50% chance of a daughter who's normally visioned, 25% chance of a son who's normally visioned, and 25% chance of a son who is colorblind. Do you see where I'm getting this ratio from? So we split it down the middle, right? Men, male offspring here on this side and female offspring on this side. Here you can see there's no possibility for the female to have if the male parent is normally vision, there's no chance for the female offspring to have color blindness. They could be a carrier still, sure, but they won't have color blindness. Here on this side, you can see that there's a 50-50 chance if the woman's a carrier and the man does not have it, there's a 50-50 chance on whether or not the son will actually have color blindness. So that's basic sex linkage for you. And so we're actually going to end off with we're going to end off excuse me. Sorry, we're going to end off with a quick look at codons and how to read codons and how to translate and whatnot. So let's just come up with something, right? So let's say our DNA sequence reads 3 prime A T, and then we're just gonna right. Okay, so this is our mRNA sequence, right? So for this, so sorry, not our mRNA sequence. This is our DNA. So we're going to translate. So we're going to transcribe from this. So first, we're going to transcribe, right? So let's first look. Okay, well for transcription, right? So transcription does it and is there's. I'm sorry, I didn't really plan this very well. For transcription, if we have an A in the DNA, we transcribe that as a U, right? So we do the opposite base pairs, right? So in this case, we have U U A C. Oh my god, I'm really sorry. This is gonna take forever. Okay, I'm sorry. You really think I'd be good at this by now, right? But it's not quite the case. Oh my god, this is pathetic. I'm sorry, guys. I'm really sorry. Let's use a shorter example because this is really sad. I'm sorry you had to watch that. So now if we do opposite base pair rules, we get the following, right? Because remember, we always oh, I messed something up here. Whoops. Sorry. Because we always pair with the opposite, right? That's base pair rules. Always pair with the opposites. I think I might have added an extra one in there, but it doesn't really matter. Oh, well, it does matter. I should have said it. I should have said a better example. Sorry. Nope, that's correct. Okay, so this is our DNA sequence. This one's our mRNA that we just transcribed, right? So now let's split it up into codons. Yes. Okay. So okay, for starters, we have to pay attention to where our start codon is, right? So it's always going to be AUG, right? And then our stop codon, right? We always have to pay attention to that first, where it starts and where it stops. So let's look at here. Where do you see AUG in this sequence? Right? So we transcribed from here. Where do you see AUG? Right here, right? So then we're going to start translation here and then we're going to end here. So this is our sequence. This is our actual transcribed sequence and then we're going to put that here. Right? So let's grab our codon chart. So this is our codon chart, right? So how do you use this thing? So basically, you start here. Okay, well, you start here, and then you just go as you see it, right? So in the case of this one, AUG, okay, we start with A, and then we go to U, and then we go to G. So that's our start codon, methionine. It's always going to be our start codon, right? So methionine, and then CUG. What's CUG? So start at C, go to U, go to G. That's leucine. I'm just going to do it in red. Leucine. Right? What about CUA? What's CUA? Okay, start at C, U, A. Leucine as well. So notice how there are redundancies in the codon chart, right? That's very important for mutations. And I have to go over mutations as well. Sorry, forgot about that. And then UAA, which is a stop codon, right? So then we just stop here. So right, so UAA, stop codon. So that is basically how you translate and transcribe a DNA sequence. Transcribe a DNA sequence, translate an RNA sequence. For the RNA sequence, for where you always have to look for where the start codon is. right? And you also have to look for where the stop codon is. So now we have mutations. So there are two types of mutations. Two types of mutations. There's frame shift and single nucleotide. So for the case of single nucleotide, we have silent, missense, and nonsense. For the case of frame shift, we have point addition and point deletion. So really quickly, point addition is, let's say we have this DNA sequence and we just added a random A here, right? So everything after this point will be affected. So this will change all of the codons following it. So that would be really bad. Same thing for a point deletion. So let's say we actually deleted this C here, right? Let's say we actually deleted that. So now everything's going to move this way, and it's going to change every codon after it, right? Which would be also really bad. So these frame shifts are the ones you have to really watch out for. These are really dangerous. As for the single nucleotide, silent. So in the case of silent, right? So remember here we had CUA, right? So CUG and CUA. So let's say in the case of CUA, this A right here actually was a G, right? It mutated to a G instead of an A. So that would actually code for the same amino acid, would it not? It would still code for leucine. So nothing actually changed in the protein expression, in the gene expression. So that would be silent mutation. So yeah. Missense mutation is if we actually changed, instead of CUA, well, if we changed U, now U is a C, so CCA. Well, let's look at here. CCA, that's proline. So now this amino acid would change to proline. That's missense. When we change one nucleotide in one codon, and now that codon codes for a different amino acid. One singular amino acid. That's missense. Now, as for nonsense, well, we can't use this example, right? But for nonsense, right? Let's use a different example. So let's say we had like AUG, CUG, and then UAC, UAA. So normally UAC codes for tyrosine, right? But if we change this last one to say UAA, well, we're actually stopping sooner than we normally would. So this would be nonsense. If this mutated to an A to say UAA, this would be a nonsense mutation because we're stopping earlier than we should. So that was mutations and how to transcribe. Oh, sorry if I just blinded you. Okay, so that's everything, right? That's pretty much everything you have to know for the exam. A good hour and 20-minute review that we just went over. I hope this was helpful. I'm sorry if it just seemed like I was rambling at points. I'm sorry if some parts weren't as helpful as I would hope. I don't know. I was just going over everything you guys had learned to this point. In any case, I wish you guys the best luck on tomorrow's exam, and I wish you guys a very happy summer. So I bid you all farewell. Bye.